final topic in our question of how cells differ is this concept of clonal expansion. The previous two topics, epigenetic and expression, talk about how cells are different and how this allows cells to perform many different functions. Clonal expansion is less about how cells can do different functions and more about some nuanced differences between cells that you may not be aware of. And so, if I were to ask you a true or false question, all somatic cells have the exact same genetic material. According to Bio 207, you would probably answer true, right? Well, in the case of clonal expansion, this is actually false. This is due to the phenomenon that mutations occur in somatic cells, and so there are these very nuanced differences between cells in our body. So for example, let's have a look at a cell as it progresses and develops. Let's say, for example, this is a zygote. It has to produce more and more cells. And in this first division, we can see it develops a mutation, marked here in red, either through the replication machinery or whatever. But a mutation here marked in red. And so then this cell goes to do further replication. What we see is its daughter cell is genetically identical, but another daughter cell has accumulated this other green mutation. And so even though these cells are present at the exact same time and reproduced by the same mother cell, as we would call it, they are slightly different from each other. So this is what we're referring to, is that cells within our body are different from each other due to mutations that accumulated. And if we extend out, we see the concept of somatic clonal expansions a bit more clearly, being that whenever you take an image of a tissue, for example, let's have a look. This would be a representative image of a tissue here, because all of these cells are in the past. These were precursors. Now all that's left in this tissue are these cells. That in general, that they're all very similar genetically, but you can see that some are more similar than others. For example, these cells here all have the exact same mutations, which is slightly different than the mutations of this cell and slightly different from the mutations in these cells. So each of these groups is referred to as a clonal expansion. Here you're seeing lineage. They're very similar words, but lineage also refers to the history of the cell, whereas expansion is more which groups are genetically identical. So we can see that there are groups here that are more similar than others just by sheer chance, phenomenon we call drift. A very similar occurrence happens in cancer cells. As we can see, just as before, cells are dividing and dividing, accumulate mutations until the first cancer-causing mutation occurs here, which results in very high level of division. And so in the end result, we see that most of the cells contain this cancer mutation here because it gave that cell an advantage. It allowed it to divide very, very quickly. So this is how tumor arises. It begins with a couple mutations that then rise and divide uncontrollably. However, it's important to understand that just because a cell has a cancer mutation doesn't necessarily mean it has cancer. And so let's visualize some of these mutations and clonal expansions through experimental results. What we're looking at here is 3 different individuals that had their esophageal epithelium sequenced. And it's important to note that these were healthy individuals with no esophagus cancer. Each of these squares represents about a square centimeter of normal esophagus tissue. And what's pictured here in these circles are different types of mutations in genes pictured here. So an orange cell has a mutation in the TP53 gene, and over here we see cells that have mutations in the Notch3 gene. And so what we're seeing in the 24 to 27-year-old is that they have mutations in many, many genes throughout their epithelium. So even though they are relatively young, they still have many mutations, and we can actually visibly see that, oh, this is a clonal expansion, here's a clonal expansion, these cells are different from those cells. And what we see is as individuals get older, these are different individuals, but we can see in the older individuals, they have more mutations. The cells are generally more mutated. Another interesting fact is that all of the mutations listed here in the colors are related to cancer. So even in this young individual, they have cells in their healthy esophageal epithelium that are cancer-causing genes. This might be confusing. It's a healthy individual, but they have cancer mutations. This is just to further our point that just because you have one cancer mutation does not mean you have cancer. And so the story of how healthy cells turn into cancer is a bit more complicated than just getting mutated in one cancer gene. Just be aware that even in your bodies right now, you are very likely to have many cells with cancer mutations in them. That doesn't mean you have cancer. So this is the concept of clonal expansion. Like I said, it's not that cells are extremely different from each other, but this is a single mutation. So keep in mind that whereas this somatic clonal expansion is technically different from this group right here, it's most likely just a few nucleotide differences, whereas we know that we have about 3.4 billion nucleotides, so still extremely genetically similar, but some mutations result in very nuanced differences between your cells.